Principles of Inheritance and Variation. Session 12. In this module, we are going to learn about the reason for incomplete dominance. In the cross conducted in Snapdragon, the phenotypic ratio is 1:2:1, which is different from the pea plant, whose ratio was 3:1, 3 dominant is to 1 recessive ratio. In the case of incomplete dominance, both the genotypic and the phenotypic ratios are the same. That is 1:2:1. Thus, when a dominant allele does not completely mask the phenotypic expression of the recessive allele in a heterozygote, a blending of both the dominant and the recessive traits takes place in the F1 and the F2 generation, and this phenomenon is called as incomplete dominance. And this is the reason why the phenotypic ratio obtained in the experiment on Snapdragon flower is different from that in pea plant. The gene for flower color controls the amount of pigments in the petals, and each allele codes for a trait, that is, specific amount of pigment production. When both the alleles for a pigment are present, the color of the petals are dark red due to heavy pigment production. Whereas, on the other hand, if none of the alleles for pigment exist, the flower is white. However, if only one of the allele is present, only half of the pigment is produced, creating a pink shade. The alleles, capital R and small r, are exhibiting incomplete dominance. The allele capital R is not completely dominant over the recessive allele small r. But why is that? That some alleles are dominant, whereas some are recessive alleles. Let us understand this by studying the functioning of gene in detail. A gene is a unit of heredity. It occupies a fixed position in a chromosome called as locus. And a gene has two forms of alleles. In homozygotes, the alleles are similar, whereas in heterozygotes, the alleles are not identical. Alleles may be different due to the changes it has undergone, which modifies the information present in that particular allele. For example, let us consider a gene that has the information to make a particular enzyme. And this gene has two copies, which are the two allelic forms. Now let us consider the normal allele produces a normal enzyme that is needed for the conversion of a substrate S into a product. According to assumed facts, the modified allele could be responsible for producing a normal or a less efficient enzyme, or a non-functional enzyme, or no enzyme at all. If the modified allele produces a normal or a less efficient enzyme, we can say that the modified allele is equivalent to the normal or unmodified allele. That is, the modified allele will produce the same phenotypic trait, or in other words, it will bring about the transformation of the substrate S. Such equivalent allele pairs are quite common. Whereas, if the modified allele, it produces a non-functional enzyme or no enzyme, then the phenotype is dependent on the unmodified or the normal allele. And here, the normal allele that produces the original phenotype is the dominant allele, and the modified allele is the recessive allele. In this example, we had just seen the recessive trait was due to a non-functional enzyme or because no enzyme was produced. That means incomplete dominance causes a distortion of the normal phenotypic ratio and results in an offspring with a combined phenotype. Dear children, in this module, we learnt about incomplete dominance and we also learnt that why alleles are dominant and why some are recessive.